All right, so chapter 6 is about infection, and we are going to spend a little bit of time sort of reviewing microbiology. If you've never taken microbiology, then this will be your introduction to microbiology. But microorganisms are basically organisms that are so small that they're visible only with a microscope, and they're so interesting that there's a whole field of study dedicated to them. We live in this weird balance with microorganisms. Some of them are very beneficial to us, and we'll talk about that. And some of them are opportunistically pathogenic, and some of them are just out to get us. So we're going to start with just a little survey of different types of microorganisms. Then we're going to talk about the principles of infection and the physiology of infection, and how to avoid it as much as can be. So we're going to start talking about bacteria. So the learning objectives for really the types, for all the different types of microorganisms, I want you to be able to list the different types and give an example of each, and to be able to briefly describe the distinguishing features of each type. And we'll go through that in the lectures. Which types of microorganisms are obligate parasites? In other words, they require a host to survive, and which are not, which are freelancers that don't require a host to survive. 4 different ways that antibiotics can kill bacteria. That's a good thing to know. Do antibiotics kill viruses, fungi, or protozoa? Why or why not? And what do the normal flora of our bodies do to help prevent disease? And under what conditions may they cause disease? So we're going to talk about all those things. But we're going to start with just a little review of microorganisms. I really liked microbiology when I was taking it in school. It's just fun to, you get to like culture and grow different microorganisms, and you get to figure out how to kill them, how you identify them, and you look at stuff under the microscope. So, that's just right up my alley. So microorganisms are just super small living forms. They're so small that you need a microscope to see them. They include bacteria, fungi, protozoa, viruses, and some other weird little ones that we'll talk about. Many of them can grow in an artificial culture medium, but many of them cannot, and we'll talk about which ones can. There are, in terms of how we relate to them as humans, there are non-pathogenic microorganisms, and then they're pathogens. So the non-pathogenic ones usually don't cause disease unless conditions change. Some of them can then take on a disease-causing role. A lot of them are part of the normal flora of our body. They live with us on a day-to-day basis, and they provide some services to us that are pretty beneficial. And so often these non-pathogenic microorganisms are beneficial. Pathogens are just flat out disease-causing microbes, and those are the baddies. We want to avoid those guys. So bacteria are part of our normal flora, and then they can also be pathogenic disease-causing organisms. They're classified as prokaryotes, which, if you recall way back in biology, prokaryotes don't have a nucleus in their cell. They're single cellular organisms, and their DNA is just floating around in the cytoplasm doing its thing. Eukaryotes, which we are eukaryotes, our cells have nuclei, and our DNA is contained within the nucleus. So there's no nuclear membrane for a prokaryote, no nucleus. They function metabolically, and they reproduce. They usually reproduce by binary fission, just dividing in 2. Bacteria have a complex cell wall structure, which is an interesting part of their defenses and offenses in the world. Most bacteria are independent. They don't require living tissues to survive, so they're not obligate parasites. They can just hang out on their own, and they vary in size and shape. So binary fission, basically, the DNA duplicates, the cell divides into 2 identical daughter cells, And then each daughter cell continues to divide. So basically you just duplicate your DNA and divide, split up the mitochondria and the ribosomes, everything else that's in the cytoplasm, and then you have identical daughter cells. The only time they are not identical is if there's some sort of mutation during the DNA duplication. And really, though, bacteria are pretty hardy because of this reproduction by binary fission. The major groups of bacteria are classified by shape. So bacilli are rod-shaped organisms. So this is an artist's rendition. This isn't what bacteria really look like, but I think they're pretty in this picture. I mean, they do look like that under the microscope, but they don't have such bright, vivid colors. So it's the color that's the artist's rendition. So the rod-shaped ones are bacilli. And there are lots of examples of those, E. coli being one of them. They're part of our normal flora, and they can also be disease-causing pathogens. Spirochetes include spiral forms. There are lots of different spirochetes. Spirochetes is the other pronunciation. Really, in terms of pronunciation, I don't care how you pronounce things as long as you know what you're talking about. And then cocci are spherical forms. So it can either be diplococci, which is two spheres together, streptococci, which are multiple spheres in a line. So streptococcus is the thing that gives you strep throat and other illnesses. Staphylococci kind of hang around in clumps. And a lot of our normal flora on our skin is Staphylococcus epidermidis. And Staphylococcus aureus is also part of our normal flora. And so it's hanging around all the time. And if those get in the wrong place, they can become pathogenic. So the basic structure of a bacteria, regardless of its shape, it has a rigid cell wall that protects and provides a specific shape. So the shape of the bacteria is ultimately provided by the cell wall. There are two types of cell walls that differ in chemical composition, and they're classified by how they take up stain underneath the microscope. So gram-positive bacteria and gram-negative bacteria. It's useful in selecting appropriate antimicrobial therapy because gram-negative bacteria have certain properties and gram-positive bacteria have other properties. And the way antimicrobial drugs work to kill them is going to vary depending on whether they're gram-positive or gram-negative. That can be determined in the laboratory very easily just by staining them with the gram stain and see which ones take up the stain and which don't. There's a cell membrane located inside the bacterial cell wall. Just like with our cells, we have a selectively permeable cell membrane. So that's one difference between bacteria and us, is we don't have cell walls. Plants have cell walls that help provide structure to their cells, but we don't. Bacteria do have cell walls, and their cell walls are a big protection device, and it has other features that help the bacteria do what it needs to do. So sometimes there is an external capsule outside the cell wall or a slime layer. It offers additional protection by keeping other organisms from getting to the bacteria. You might have flagellae, which are little protuberances that are attached to the cell wall, and they can almost be like a little tail to provide motility for some species that live in a fluid environment. They can paddle around with the flagellae. They're little hair-like structures called pili or fimbriae that are found in some bacteria. They are along the edge of the cell wall, and sometimes they assist in attachment to tissues or surfaces. And they also assist in the transfer of DNA to another bacterium. So even though bacteria generally reproduce by binary fission, they can conjugate and exchange genetic material. And often that genetic material that is exchanged is a resistance to antibiotics. So antibiotic resistant bacteria can be a big issue. And if bacteria are conjugating, they can transfer their resistance to another bacterium. So the cell membrane inside the bacterial cell wall is selectively permeable, just like our cell membranes. The cytoplasm contains one long strand of DNA, just one chromosome, instead of multiple ones like we have. It contains ribosomes and RNA and plasmids. And plasmids are the DNA fragments that are non-chromosomal, and those are what are exchanged during conjugation. And those are usually giving the bacteria some antibiotic resistance. So a plasmid is a little circle of DNA that can be transferred to another bacterium. So bacteria also sometimes produce toxins. So they're exotoxins, which are usually produced by gram-positive bacteria, endotoxins, which are present in the cell wall of gram-negative bacteria, and they're released when the bacterium dies. And the endotoxins can be vasoactive compounds that can cause septic shock when you have an infection from these gram-negative bacteria. So that's really serious. That could kill you, actually. Bacteria also have enzymes which can damage tissues and promote the spread of infection. So when we're talking about specific infection and the physiology of infection, we'll talk about how this works. But so bacteria seem like, oh, happy little guys, they're going along, you know, making cheese and fermenting wine and whatnot, but they are also, well, I guess it's yeast that's fermenting the wine, but they can be pretty bad little guys. They can kill us, so we got to watch out for them. Some bacteria can form spores. So a spore is a dormant or latent form of a bacterium, and it can survive in long periods of time in a spore state. They're highly resistant to heat and disinfectants. So that's not good. Or, you know, it's good for the bacterium. It's not necessarily good for us. So antimicrobial drugs, which we'll definitely talk more about, they target certain structures in the bacterium. So because bacteria can acquire antibiotic resistance and antimicrobial resistance, we have to keep, our pharmacologists have to keep coming up with better ways to kill bacteria, right? So one way they do it is to target the bacterial cell wall. So penicillin, it was one of the first antibiotics discovered, and that's what penicillin does. It targets the bacterial cell wall. So it breaks open the cell and kills it. You can also have antibiotics that interfere with the metabolism of the bacterium, and those sulfonamides is one example of that. You can have antimicrobial drugs that interfere with chromosome, DNA, and protein synthesis, like tetracycline. We'll talk about things that are bactericidal or bacteriostatic. Bacteriostatic just makes it so that they don't really grow and reproduce. It doesn't necessarily kill them. But it eventually kills them off because they don't grow and reproduce. There are also drugs that cause cell membrane damage, like polymyxin. So because bacteria keep acquiring resistance, we have to keep coming up with new ways to fight bacteria. So there is definitely a problem with too much use of antibiotics, where the bacteria are just going to adapt to it, and we're going to have worse and worse things in order to kill them.